Hello everyone, this is a new video for my YouTube channel. This is about descriptive statistics on categorical variables. Until now we have gone through understanding the main descriptive statistics for numerical variables. Now we are going through the categorical variables, so just to explain the basics function that we can find in R to describe those type of variable. First of all, loading the data. This time I use a different data set from the previous one. Just for everyone who is not familiar with R has several built-in data sets, and by simply clicking data followed by brackets, you will have a list of built-in data set that you can easily find in R and you can use to learn or just to analyze. You can see here there are quite a few data sets of every different type. Today we're using this data set here. It's carbon dioxide uptake in grass plants. And it's called CO2. So I load the CO2 data set. That was my mistake here. So then we see, as usual, we're going through the data set, trying to understanding what it is. We know we have 84 observation and 5 variables. What are these variables for? The variables are we have factors, ordinal factors, normal factors with 2 levels, and factors and other factors with 2 levels. So the good function of str function is that provides you with a lot of information about those variables, basically the name, and also it will tell you if the variable is ordinal or not. Factor in R, it assumes to be a variable which is categorical. In this case we have a factor with 12 levels and another factor with 2 levels and a third one with 2 levels, plus 2 numerical variables which we will see later on. If we run the head function, you can clearly see the structures of this database, the first 6 observation, and we do have plant type, treatment, concentration and uptake. So first things we can do is sorry, to summarize the data, as you would always do in R. You can use the summary function. If you remember from previous videos, first you put the name of the data set followed by the $sign and the name of the variable. So we are concentrating on the first 3 variables, which are factors, and if we see here by simply running summary, you'll have for each specific level of your factor the number of observation that satisfy this factor. So for here we have 12 levels, as we already know. Each of them has 7 observation. If we click type, we know from the previous command that we are expecting 2 levels, and in fact this is the case. We have 2 levels, the levels Quebec and Mississippi, and there are 42 each. Same would apply to treatment, and again is just exactly the same, non-chilled and chilled, and again 42 each. One way to describe a categorical variable is to use the function table. So table would provide you with a structure results, which is very similar, as you see, from the summary type of results. But table can also allow you to do multiple calculation on this data. So let's say you want to see the treatment table. I have created a new table here, which is called table 1 with the function table. So with this sign you can basically create a new object inside your environment. In this case, I'm creating an object called table1, and I want it to be the table of the variable treatment. When I run this, the output is basically none. In fact, the output is inside the new object. So to report the output, I have to type the name of the object, and there you go. It's very similar to the previous table, but this time for the variable treatment with the two levels reported. There's a function if you can want to see the summary of this, which is called add margins, and in this case for table 1 you can see the margins. The add margins function is reporting you the sum of this table. But what if I want to do a percentage of this table? If you run prop table, this will provide you with the percentage. Prop stands for proportion, and if you run this, you clearly see that 0.5 and 0.5 are the percentage. Of course, this means the overall number will make 1. If you want it by proper percentages, meaning in a scale from 0 to 100, you multiply this 400 and you have 50 and 50%. Even here you can introduce the add margins function for the table in percentage, and you will have of course a sum which is 100%. If you want to describe two categorical variable, one crossing the others, say you want to know the type by treatment, here I have created a new object called table2. In this case, you put table, the first variable here, then a comma and another variable here, and you simply obtain a cross table. Here we know that they are equally distributed. We have 21 non-chilled in Quebec and 21 chill in Quebec, and the same apply to Mississippi, 21 and 21. Even here you can add margins as before, in which you'll have the sum on both column and row, and this is the overall sum of everything. So, but what if you just want the columns? You have to introduce in add margins, you have to introduce the number 1. So if you type this, you have the summary only by columns. If you put the number 2, you'll have it by rows. So by typing question mark add margins, you will have the information of this sequence and how to change those details. Every time in R you want to have some information about a specific function, you just type the command and the name of the function. The question mark and the name of the function. So now we know that basically we have 21 non-chilled and chilled for each of the type Quebec and Mississippi. If we want to plot, one important function in R is the bar plot function, and this is a very interesting type of plot for categorical variable. First of all, I just use the table 2 which I have created, so it's a cross table. The X lab stands for the label of the X axis, and of course in this case the X axis is the treatment. The Y lab is the same for the Y axis, in this case is a frequency because we have the overall number of events for each variable. The main in R plot is the name basically of the main title of the entire plot. There are several different ways to name each specific part of a plot, but main is the main title. Col stands for color, and I decided to color them light green and light blue. And then of course the legend, you have to put the legend obtained from the names of your table. So by knowing that the row names here are referring to the type, you simply say row names table, Table 2 is actual our tables. And the arguments for legends, in this case, were to put my legends, and I decided to put it on the top right part of a plot. If I click all of this, this is what I've obtained. You can clearly see here that we have non-chilled and chilled. We have 21 per group, as we know. The overall is about 42. Surely we have a legend here telling us which one is which, Mississippi and Quebec, and of course you can clearly easily find out that are equally distributed in this case. What if I do the percentage of this table too, and again I obtain a very similar data to the previous one. You can clearly see here we have 25% for each group, as you would expect. Now I can create a new table. I will call it table 3 in this case, and the proportion of table 2 basically with a real percentage. In this case, table 3 is 25% each group, and we know that. I will add margins, and the sum it's 100%, so everything looks correct here. I can rerun the plot. In this case, I use table 3 as I am plotting the percentages. Of course, I changed the YLab as a percentages. I kept the same colors, the same row names table 3, the arguments legends top right as before. If I run this, a very similar plot, but this time you'll have the percentages on this side. So here, as you would expect from the table, you have 25% of the table, and the overall total is 50%. Again, the legend is very similar. The two groups are exposed here. So, but what if I want to describe a numerical variable depending on a categorical variable? So if I run table, in this case I probably not obtaining what I really want. So if I plot table, you can clearly see here it doesn't really make sense. Basically, I know that the uptake of 7.7 happened in 1 chilled and 9.3, so it's basically treating the numerical variable as a categorical variable. Really doesn't make much sense if you want to describe a numerical variable. In this case, I frequently use, as you've seen before, a library psych, which has inside its function inside this package a function called describe by. Now this function requires you to first put the variable you want to analyze, which in this case is uptake, is a numerical variable, and then the group function, in which case I use the treatment function. If I run this, I see clearly that depending on the group I have a different mean, standard deviation, and so on. In the next video we'll go through how to plot all of this and how to analyze this data in a larger scale for categorical variables and so on. Thank you.